Namaskar, welcome to PK's classes. Today we are going to study the chemical method of gene sequencing. There are two methods of gene sequencing. One is the dideoxy chain terminator method and the second is the chemical method of gene sequencing. And this chemical method was developed by Alan Maxam and Walter Gilbert, and accordingly it is named as Maxam and Gilbert chemical method of DNA sequencing or gene sequencing. So when you say gene sequencing in the DNA, which is a double helix structure and two strands are there, and each strand is a chain of sugar and phosphate, and to each sugar there is a base out of these four: adenine, cytosine, guanine and thymine, and that base you can say A, G, T, C, A, G, C, C, T, like this. The base sequence actually we know through this technique of gene sequencing or DNA sequencing. So whatever the base sequence are there in one strand, that we can know, and from that the other strand can also be known. So initially, to the strand to be from which we need to identify the sequence of genes, that is radio labeled with P32, phosphorus 32, and then the chemicals, four different chemicals are used in four different sample containers, and when we use piperidine format, then there will be break at both adenine base and guanine base, and when we use dimethyl sulfate, then there will be break at guanine, and if we use hydrogen and sodium chloride, then it will break at cytosine, and when we use only hydrogen, then it will break at both C plus T, both at C and as well as T. So we identify it as A when there is a spot at this column where we have used piperidine format. A plus G column, we will get one spot, then it is identified as A. For G, both piperidine format will break as well as the dimethyl sulfate also will cause the break at G. So we will get two spots at G and we identify it as G. Similarly, for C, we get two spots at this C column as well as at C plus T column. When we use hydrogen and sodium chloride, there will be break at C. When we use only hydrogen, there will also be break at C as well as T. So we will get two spots for C and we will identify it as C. But when we get only one spot at A plus G, then it is identified as A, and when we get only one spot at T plus C, then we will identify it as T, and accordingly we will know the base sequence this way. So C, T, A, C, G, T, A. So you can see here C, T, A, C, G, T, A. This is the sequence which we have already initially taken. This is the DNA single strand which is unknown and we want to identify it. So we have radio labeled with phosphorus 32. Then the chemicals are added and there will be breaks, so we will get DNA fragments of different lengths. So based on the size, we will run them in the gel electrophoresis. The larger one will remain at the bottom and the smaller size one will move upwards, and then we can get the sequence of the DNA strand. So let us summarize now. So it is radio labeled followed by base specific cleavage. In the A reaction there will be some cleavage at G. In the T reaction there will be some cleavage at C. and we will do the electrophoresis followed by autoradiography, and then we can visualize the base sequence, and accordingly we can know the gene sequence or DNA sequence. So the chemical method is a very sensitive method, but it is less frequently used nowadays because the chemicals are toxic. The chemicals are toxic as compared to the Sanger method. And the second thing is these methods are labor intensive as compared to the Sanger method. However, for small DNA fragments like oligonucleotides, chemical method is suitable, and the primary advantage of the Maxam Gilbert or chemical method of sequencing over Sanger method is that DNA sequences are obtained from the original DNA molecule and not from a synthesized copy. So thus one can analyze the DNA modifications such as methylation and study DNA secondary structure and the interaction of proteins with DNA, all using this Maxam Gilbert method. And the points you can note down. We have already discussed the difference between these points of Maxam Gilbert sequencing and Sanger sequencing. So this is all. Thank you.